Hello, everyone, and welcome to the July webinar. This is Rebecca Bailey, and I will be the moderator for today's webinar. We do have one housekeeping item today. I just want to let everyone know that you can ask questions of our presenter by entering the questions into the questions pane on the GoToWebinar side panel. Today's speaker, we are getting a bit of an echo. Marcus, can I have you mute for just a moment? Thank you. So today's speaker is Dr. Marcus Rosenblatt, and I am hoping I pronounced that correctly. Marcus is a postdoctoral researcher at the University of Freiburg in Germany, where he works in the group of Jens Timmer on data analysis and modeling in the life sciences. He received his bachelor's degree in physics from the Freie Universität in Berlin, and his master's degree from the University of Freiburg, where he developed an interest in studying biology at the systems level. This interest has led him to pursue a PhD in systems biology, also at the University of Freiburg, where he specialized in developing and refining methods for parameter estimation in ODE models. He applied these methods in various collaboration projects, both within Freiburg and in the group of Ursula Klingmüller at the German Cancer Research Center in Heidelberg. Marcus has also completed 2 internships during his PhD, one at the Walter and Eliza Hall Institute in Melbourne, Australia, and one in Interferon and Basel, Switzerland. In 2022, Marcus was awarded the MTZ Award for Medical Systems Biology, recognizing his outstanding contributions to the field. So with that, I would love to give a very warm welcome to our speaker today. Welcome, Marcus, and we are looking forward to this presentation.
Thank you, Rebecca, for this nice introduction. I will switch my camera off for the connection and then start screen sharing. Yes. So yeah, today I want to talk about the mechanistic insights into sensitization and desensitization of interferon alpha signaling and its effect on patient treatment strategies. And just a short outline, I will first speak about this ODE modeling project and also introduce or show you some methods there that are usually applied in the tumor group in our lab here in Freiburg. And then later I will talk about some applications to human data, patient-derived human hepatocyte data, and also talk about pharmacokinetics and treatment strategies. So interferons, yeah, why and where they matter. So they are pretty much involved in a lot of things, yeah. So they are also a very current topic, especially since the corona pandemic. They became more of interest because timing and localization of interferon response seem to have an influence on the curing chances of after a SARS-CoV-2 infection. And so in general, interferons are involved in the antiviral response, inhibition of cell growth, cell death, and so on, and also it's an anti-tumor activator. And so when you apply them therapeutically, which is also done, for example, against HPV or HCV infections, some patients do not respond, which is a major problem. And the signaling pathway that is connected to the interferons is known to desensitize upon activation. And we wanted to, or we built a ODE model to understand this mechanistic origin of this desensitization. So let's look a little bit deeper into the pathway. And how does it look like? if a virus gets sensed by the cell, interferons are activated. This is what you see on the left. And then these interferons activate their own signaling pathway. This is a JAK-STAT signaling pathway. And there are three main complexes that are activated. This is the STAT1 homodimer complex, the ISGs3 complex, and the STAT1, STAT2 heterodimer complex. And they then, initiate a gene response and activate interferon stimulated genes. And so what makes the system very complicated is that there is lots of feedbacks involved in the system. There's three main negative feedbacks, SOCS1, SOCS3, and USP18, and also three positive feedbacks, STAT1, STAT2, and IRF9. And these, so STAT1, STAT2, and IRF9, by the way, I can say this already now, these are forming this ISG3 complex, which is a main readout of the pathway. So when our collaboration partners, as Rebecca said, it's the group of Ursula Klingmüller in Heidelberg in Germany at the DKFC, they investigated sensitization experiments on, with stimulating cells with interferon alpha. So what they did is they pre-stimulated cells with different doses and then looked at the response after a second stimulation. So you can see this here on the lower left in this timeline. So the measurements in this case were taking have been taken at 24 hours or after 24 hours. And what you then observe is, so in black, this is the control where you have no pre-stimulation and just one stimulation, you get a nice response of the system. But if you pre-stimulate with a high dose, this is this purple condition, then the response is essentially not there anymore. And what can also happen, this was new at this point, a couple of years ago, was that if you pre-stimulate with a low dose, you can even get a higher response of the system, which is of course very interesting. And so now, just to give you a little bit of an idea how the work in our lab is going. Usually we have some research questions, some biological hypothesis, and then there's a long way to a mechanistic and quantitative understanding of these systems of the what you investigate. And there's lots of methods going in here. I will speak about a couple of them, but this is just to get you an idea. This is really a complex process that usually goes over years in some projects, and it's always, you have to always think it in cycles, because if you have some results, you go back to the start and so on. Yeah. So, and first, let's look at the ODE model generation building process, how we do this usually. So on the left, you have again this biological cartoon that I introduced previously, and this is basically translated into a reaction network here in systems biology graphical notation, which is where essentially each arrow in this cartoon represents a reaction. And these reactions, so the rate laws of these reactions are derived, for example, or mostly from mass action. And but there's also enzyme kinetics and, for example, inhibition terms. So this is always how we set up the models. There's, of course, the details are more complicated, but this is how it works in principle. And the idea is then to determine unknown parameters of the system by maximum likelihood estimation given experimental data. And so this is on the right side how this looks like in some particular reactions that are given in this particular project here. For example, the form where some ISG3 complexes formed depending on IRF9, STAT1, and STAT2. Yeah, I don't want to go into the details here. I will focus later on specific parts of the model. So, and yeah, after we have a model, ODE model generated, of course, we want to know how this model performs. So we fit in, or we we plug in the experimental data that we have and perform parameter estimation, which is essentially an optimization of the system. So given we have this equation system, which is in this sense here called F. So you have a time derivative of the ODE model. Say it's just x dot is f of x, p, and u, and so x is the states, p is the parameters, and u is some time dependent input functions, which, for example, represent the experimental conditions of your system. Then you translate this into observable functions, and so this is this function g here. And usually our systems are only partially observed, so the dimension of this state vector y is much less than of the vector x. And then we do a maximum likelihood estimation, and if you have Gaussian noise, then this essentially boils down to minimizing the weighted residual sum of squares. And yeah, given some data, in this here it's called YDKL, yeah, and given some also uncertainties of this data, you can minimize this function. And the methodology, you need lots of things for this. For example, sensitivity equations is all implemented in 2 different frameworks that were developed in our group. For example, the DMOD framework in the language R, and then there's this data to dynamics framework in MATLAB. So now let's look at some data really that are connected to the project I want to talk about. So it's again the pathway cartoon on the left. And on the right, you see some data of these feedback proteins. So when you see, so this is qPCR data. Who is familiar with that? It's just measuring the mRNA over time. And you see that these feedbacks are induced in different ways. So some of them seem to be sustained and some are transient. And then we also have lots of Western blot data in this project. This is essentially measuring the protein states. And for example, phosphostat1, what you see here in the middle, this is basically the pathway activation, if you want so. And then there's also the protein of the feedback components measured. So, and just I cannot go into all the data that went into this model, so because it's about 12,000 data points, because this model is also, as you can see already from the cartoon, a very big model. So not always the models in our group are not always like that, but some are. Yeah, and so in here you have like 40 dynamic variables and 85 dynamic parameters, which is really a lot. And so, yeah, apart from Western blot and qPCR measurements, there's also mass spec data measured. And what is maybe interesting or different in other projects, so the time course that was used here is really measured over a long time period, in this case 32 hours, and yeah, including several experimental conditions. So, but now let's look about on the sensitization of the pathway. And I already said that if you pre-stimulate with a high dose, then the response is essentially not visible anymore. This is again this purple condition here. And here we investigated basically different pre-stimulation doses. And what you see, the dots is the data, of course, yeah, and the lines that you see is the model prediction or the model fit. And you see the model can nicely describe this sensitization data. And by means of this output of the model, we can now, of course, define a measure for the antiviral response of the system. So I just said it briefly before. So I say it again, in this phosphostat1, phosphostat2 is the main pathway readout. So it's just the phosphorylation of this STAT complexes is measured here. And this is then going further downstream to shape the antiviral response. I will be more precise in a couple of minutes on this. But you can define a measure for this antiviral response via the area under the curve of this particular model state. And what you then can do is with this different pre-stimulation doses that you have, you can draw this curve essentially, where you plot the antiviral response over the amount of pre-stimulation that you have. And of course, if you have no pre-stimulation, then it's somehow normalized to 1 here. So this is the relative antiviral response, which is just normalized to this black dot here. And then you have 2 regimes. So the hypersensitization regime, or we also say priming sometimes, where you get a higher response, and then at some point, which we call the desensitization threshold, you go into this desensitization regime, which is problematic for the clinics. And if you analyze the model, you can find out that the feedback proteins, in this case it's IRF9 and STAT2, that are relevant for this hypersensitization, and USP18 and SOCS1 are relevant for the desensitization. So now coming a little bit more to the methods part, how can we actually fit and generate such big models, and how can we find out something of them, or be sure that what we found out is correct, so to say, yeah. And there's a couple of methods that we are usually using, and that were also, lots of them were also developed in our group. I cannot speak about all of them, but I will introduce shortly a couple of them. So a major thing is that we do deterministic optimization from multiple points in parameter space. This is what you see here on the upper left. So you start from different positions, and then really in a deterministic way minimize the objective function, and eventually find the global optimum of the system. And what we then usually do is we plot this in a so-called waterfall plot, so where we plot the objective function over the index of the optimization. So in this case, it's 50 optimizations have been performed. And then if we see a step function like in the blue shape in the upper left, upper right panel, then we are pretty sure that this corresponds to local optima, and that we really found the global optimum, which is here marked with A. And there was this paper in 2013 from Andreas Rauer, where he compared also other optimizers, and it was found out that this deterministic optimization works best in our cases. What is also very important is that we analyze the confidence intervals of the parameters. And this is done via the profile likelihood. You can find it in this paper from 2009. Essentially, we classify parameters in these 3 different cases, A, B, and C, that you see. So we, by means of this profile likelihood, we can say a parameter is identifiable if he has a finite confidence interval, and if the confidence interval is not finite, then we speak of a non-identifiable parameter, and this can be structural or practical. Then going through when you have such a model, yeah, and you want to answer a biological question, then you perform several steps to tailor your model complexity to the information content of the data. And this is a little bit a statement, and there's also different philosophies out there, people approaching this in another way, but this is at least our philosophy in our group, and which works well for our cases. This is yeah, if you want to learn more about this, you have to look into this paper from 2021. Yeah, essentially, you remove all structural non-identifiabilities, and then you look at the practical ones, and yeah, and by this really make your model in a way that it really tailors to the data. To be a little bit more precise on what's meant by this, if we speak of a non-identifiable parameter, what is actually the problem with this? Yeah, let's look at this toy model. A goes to B goes to C, and now let's assume that only the state C is measured, which you can see here on the right. So we only have data points between 25 and 50. And then these green and red curves are different parameter sets and the trajectories of these parameter sets for the ODE. And then, of course, yeah, there are, for example, the green ones, they all fit the data very good. But when you now look at the states A and B, it's not clear which trajectory is the right one. So, and this is essentially visible in this profile likelihood, where we see that these parameters have a confidence interval which is open to plus infinity. And yeah, these values of the parameters, they in this case cannot really be determined, and the uncertainty quantification is impossible, you might say. And the problem with that is that the predictions of the model, and you always want to make the predictions that you can falsify, they may become non-meaningful. And this is, yeah, this is the problem why we have to, why we always look at this and find this so important. And there are different strategies to cure this non-identifiability problem. One is that you make proper experimental design, that you basically collect more data, but you design a priori by means of the model which experiments are most informative. But you can also go the other way and take your model that you have and reduce it to the complexity that you need. Yeah. This was probably now lots of input from this method side, and I was not really able to go into the detail timing wise. If you have questions on this, I'm happy to answer them later, or you come back to me by mail or whatever. You can also go through these papers. I just wanted to mention these very important things, or at least important things to my eyes. Right? Now let's go back to the project, to the interferon alpha signaling project, and look by using this model that we have there, how the predictions of this model look like. Sorry, now here. Now you see the slide, right? Again, this is on the left, the model cartoon, and now we specifically look at these signaling complexes that I already mentioned. So this is here the STAT1 homodimer in red, the STAT1, STAT2 heterodimer in yellow, and this ISGF3 complex collecting also IRF9 in green. And it turns out that for different, this is what you see on the right, for different pre-stimulations, the dynamics of these three states looks completely different. So if you look on the top where you have no pre-stimulation, the homodimers are formed earlier, then the heterodimers come up, and the ISG3 is the last one. Yeah. But if you have a pre-stimulation and then stimulate, then the situation looks completely different. And this is the important thing, that the model is really able to predict these dynamical behaviors. And I will show you in a second how we can validate this. Essentially, you take your model and you calculate, this is another approach, yeah, the prediction profile likelihood. And this is essentially, it computes you a confidence interval of your model prediction. This is what you see as these shaded areas in this right plot. And essentially what you have to look at here is that this data, and because this is these data points that you see here was data that the model has never seen, but they're really in line with the model predictions, which was nice to see. And the experimental techniques that were applied here were a little bit more complicated, so to say, and this is why they are usually not performed. But for such a model validation where you know in which particular time point and which particular dose you're looking at, or you have to look at, you can do this, and then by means of this validate your model. And this was really nice to see. At this point, we trusted that our model really does what we think. And you can also go one step further, look at the gene response, and this is essentially one step downstream of the signaling complexes. And there's this promoter sites occupy the gamma activated sequence. This is GAS, the abbreviation, and then there's this ISRA, which stands for Interferon Stimulated Response Element. And you can predict with the model also dynamics for these, given some experimental conditions, also including different pre-stimulation doses. And then by means of a promoter analysis, look at target genes that are specifically connected to these binding sites. And you see on the left, on the right, that the predictions of the model are also in line with this data that was generated after the model calibration. You can also, okay, sorry, I have to go back one slide. No. So these, again, there's the GAS binding sites on the left and the ISRA on the right. And what you see is that this GAS binding sites essentially has no hypersensitization. So you see it because the yellow and the green is essentially below the black curve. But for the ISRA, it's different. So green and yellow are above the black one. And so black is this untreated control case. And there you see that potentially there's a very strong hypersensitization if the dose and timing is chosen properly. And this is maybe one of the main messages here of this this figure. And of course, looking at dosing and time, you can also do this with the model for all time points and for all pre-stimulation doses. And interestingly, there seems to be a very nice optimal position in this space, so which is around 16 hours. So with this pre-stimulation dose of about 28 picomolar, and this just means that to get the most response out of your system after 2 doses, you need to pre-stimulate with this low dose and then re-stimulate at this particular time point, which is around 16 hours. So now I come to the second part of the talk, and applications of this model to patient-derived human hepatocytes and how we can use this for pharmacokinetics and treatment strategies questions. So we have this model, ODE model, and this was, I think I never said it during this talk now, it was all calibrated on one particular hepatoma cell line, which is named HUH7.5, yeah. And but now these sensitization experiments that I showed you, they were also performed for another cell line, which is called HepG2, and also for primary human hepatocytes. And it turns out, this is what you see on the left, that the effect with low pre-stimulation gives you a hypersensitization, and that the high pre-stimulation gives you a desensitization is conserved also in these cell systems. And then we applied the model and used this the same model structure for these cell systems, and accounted for differences between the cell systems by some specific parameters, and these are identified by means of L1 regularization. If you want to learn more about this, you can look at these two papers. Yeah, I also cannot really go into detail here. No, next slide. Yes. But the interesting thing to see was, if you remember this charistic shape that we had, the relative antiviral response plotted over the pre-stimulation dose. This is essentially the same plot here, but now for all cell systems. And you see that this shape that we observed, that we have these two different regimes, the hypersensitization and desensitization regime, is conserved over all these cell systems. And this, the exact positioning of this shape is determined by the abundance of feedback proteins. And then this is these dots that I plotted here. This is exactly this desensitization threshold that we defined. And if you now plot this over the abundance of feedbacks, you see that USP18 in untreated cells is essentially a very good biomarker for this desensitization threshold. Now, further adaptations of the model and extensions to pharmacokinetics. So first of all, I wanted to show you that what we are doing, also other people take it over. So this was interesting to see. It's also actually very recently, I think from March this year, a paper that where they, from the Hoffman group, where they actually they took our model and fitted it and took the dynamics of this ISGF3 readout that we also had and fitted it to, or used it as an input to RNA transcriptome data that they had. And then they classified clusters of these transcriptomes and linked it to special antiviral effector functions. And this is, to my eyes, essentially the way to go, how we can use these type of ODE models, for example, for signaling pathway, really to shape or to show the clinical relevance. And we actually, in parallel, to be honest, but we are doing something similar. So what they did was only for one stimulation. They looked at data only for one stimulation, but of course, our strength is that we also have this information on multiple treatments or multiple doses. And we also currently looking at in the project on measuring proteins, not the RNA, but proteins, and to link this to this transcription factor dynamics that you see here on the right. And so the idea is to this is maybe the better figure for explaining this, to take the ODE model and predict these three different transcription factors, plug it into some mini models, and essentially predict the protein dynamics. And by means of this, you can then figure out which of these proteins is activated by which transcription factor. And this is, if you remember that they have very different dynamics, is very interesting. Also, if you have some particular protein that needs to be activated in order to do some antiviral effector function, then you can really predict this with the model. This is the idea. So this is, so to say, downstream of the model, linking the model further downstream, but you also have, of course, to take into account upstream of the model, namely the pharmacokinetic effect, if you think of interferon in the body. And there we currently have a bachelor student working on this, Philip Weibel, where he essentially added some very basic pharmacokinetic model to this to our given ODE model, and looked at all particular parameters, introduced this absorption and elimination parameter, and did some prediction on this. And so this is still preliminary results that you see here, but the interesting thing is that this relative antiviral response shape is actually conserved also for this what I termed ROF and PEG. So this stands for roferone and Pegasus, which is essentially two different agents that are also used in the clinics really to given to patients. Yeah. And we can really show that this observation with this hypersensitization and desensitization also holds true for these two drugs. And yeah, by means of this PK model, you can now, of course, also look at the influence of these pharmacokinetic parameters. And yeah, again, this is preliminary results, but it's interesting to see. So this is again this what you see in the different panels is again the heat map. So where you have on the x-axis the time point of the second stimulation, and on the y-axis the dose of the first stimulation. So, and if you remember, there was this one particular optimum that we found, and this is now plotted for different values of this elimination parameter and of this absorption parameter. And interestingly, there seems to be an optimum at this 0.5 value, and so this is interesting to see, because this is very much in the physiological range in the body. And so this is plotted here for HGH 7.5. This is again this cell line where the ODE model originally was calibrated on. But you can now, of course, also do all these kinds of plots for the patients that we have measured. And you see that it looks a little bit different there. And for example, it can also look like this. Yeah. So this is just, as I said, so this is what we are currently doing, and trying to figure out by means of such analysis which parameters in the patients are relevant for which things, and how can we use this knowledge for optimal treatment scheme. Yeah. And yeah, with this, I already come to the summary. I was a little bit faster than I thought, but it's also good. So we have this dose-dependent sensitization. This is observable across multiple cell systems, including also primary human hepatocytes. Different stimuli, including their pharmacokinetics, also for these, we always see the same shape. So this effect is really conserved. And we see it on the level of transcriptive factors, we see it on the level of the promoter regions, and on the interferon stimulated genes themselves. And this is work in progress. We will, I'm pretty sure on this, also see it on the interferon stimulated proteins. So meaning not mRNA, but protein. Yeah. And now, if you think about patient individual optimal treatment, this is, yeah, it's still hard to say what is really optimal, but we already know that it's influenced by the abundance of feedback proteins of the system, by the PK parameters, and it also depends on which readout you're looking at. And this is interesting if you think on also a clinical perspective, where you know that you need, as I said before, one specific protein that needs to be upregulated, and you have first to find out whether this is more activated by this ESRA element or by the GAS element, and with this will also influence the optimal treatment that you have. And yeah, but what's also interesting to see this optimal treatment time point, where I said it was like at around 16 hours. It's different if you have different pharmacokinetics, but it's relatively stable. That it doesn't depend on the other parameters of the system, to phrase it like this. Yeah, and of course, next we would like to optimize this response also with multiple treatments, because this is then even one step further into the clinics, and compare it to the clinical application that is really done. And yeah, we are still struggling with the question whether we would need more patient data for this or not. This is always a little bit complicated if you are in academia, but yeah, this is how it is. With this, I want to thank all the people involved in this ODE modeling project. Yeah, this is the group of Ursula Klingmüller that I already mentioned, and of course also all my colleagues in the Timber group, especially Philip and Jack, and yeah, some other groups that contributed to the data generation for this ODE model. And of course also the funding agencies on the right. And I thank you for your attention.
Thank you, Marcus. I'd like to just remind our audience that you can ask Marcus questions by putting them into the questions box on your GoToWebinar side panel. We do have some questions, so I'm going to start with so Marcus, you modeled the downstream signaling of the interferon receptor in much detail. However, the receptor dynamics also exhibit some complexity involving assembly of multiple subunits. The questioner is asking, I'm wondering why you decided against a more detailed representation of the receptor dynamics.
Yeah, that's a good question. We also had lots of discussions on that. Yeah, the answer is basically in this thing with tailoring the model complexity to the information content of the data, because basically we do not have data on the receptor level, on not precise. So this or at that time when the ODE model was built, we didn't have data on this. This now starts that we can also measure this, but it's still complicated. But so if whoever asked the question has more also data-wise information on this, this would of course be interesting. Yeah, then you could add this to the model very easily. Yeah.
Our second question, we have a couple of questions, and I'm going to sort of combine them because they're very similar. They're asking about how the interferon induction was measured and whether or not your research would have been different had you picked a different outcome measure.
Yeah, so well, the outcome measure is essentially was always defined via the pathway readout. So in this case was for STAT1 and 2, and they were both pretty similar in the first place. But yeah, it's true that if you look at the other readout, then the results that you get are also a little bit different. But it's interesting to see that this, especially these two regimes with the sensitization and desensitization, is really conserved no matter which readout you're looking at, or at least if you look at a meaningful readout, then it's always conserved. Yeah. I'm not sure. I hope this answers the question. Yeah.
Okay. You said you started with the first cell type, the HUH cells, and then converted to the other cell types. How different are the models between the cell types?
Yeah. This is interesting. Yeah, I cannot say it by heart, but so let me go to the slide again. So essentially, what you try is that you with this L1 regularization method is that you really try to have as many parameters in common between the cell systems and only figure out the ones that are really relevant for the other cell types. And if I remember correctly, it was 2 parameters for the difference between HUH7.5 and compared to HepG2. And it was, I think it was 4 to bring it to the primary human hepatocyte. But I have to mention, in what was always assumed to be different is the abundance of feedback components. This is what you see here on the lower left. And these were also measured, for example, in this patient-derived human hepatocytes. So these 4 parameters were always different, but adding to these, I think it was 4 more. Yeah. But I would have to look into the paper myself. So, yeah.
Could these be linked to a specific pathway that would be different between the cell types?
These parameters, yeah, I mean, I'm not sure if it would be possible in our case because I'm not sure. Actually, I'm sure that we would not have enough data for that. But in theory, yes. And there are also other projects doing this where you can really find out this and this parameter needs to be different, and then they found out, okay, there is this and this mutation which actually explains this. And this is also the idea behind, yeah, that really you can figure out this needs to be different, and then look more into the biology, what is the reason for that, and to understand it in this sense. Yes.
Thanks. For the audience that are asking, yes, we will be posting the slides and some of the references on the website after the webinar. So one of the questions that came up was that liver, the data is from a cell culture, and that livers in humans are composed of different cell types. Would you expect additional feedback if you included input from those other cell types?
Yeah, so this is true. Yeah, this is mostly in the liver side. Yeah, if you would use another cell type, I'm not sure if there are other feedbacks there. I have not enough experience, to be honest. We also with one of our colleagues here in Freiburg also thought about doing some similar investigations on, for example, T cells, but this was never done yet, at least to my knowledge. I think you would not have additional feedbacks, but the parameters still would be different. Yeah, and this can, for example, then of course also shape the response in a different way. But so for the pathway components and the main feedbacks, I would think that it's the same, but I cannot say for sure. Yeah.
So one of the audience would like to know, did you measure the interferon alpha dynamics in these experiments? Is the interferon alpha consumed faster by a receptor-mediated pathway?
Yeah, that's an interesting thing. Now we didn't measure it in, so at least we didn't collect data for ourselves, but in this, let me go to the slide. Oh, this was too fast. Here, so these parameters that went into these PK plots of Rover and Pegasus, they were taken from literature. Yeah, so I didn't cite all these references here because this is really a lot. You have to essentially look into the publications of Rover and Pegasus, yeah, and you find out there. But so, and of course, in these publications there was also data, but we for ourselves didn't measure it. No.
So with the desensitization, did you also look at how long it takes for recovery of the cells?
Yeah. So I'm not able to show it because this would go, I have to dig it out somewhere, but I have once done it, and it seems that, so at least for interferon and HUH7.5 that it takes like 2 or 3 days after some high stimulation till the system relaxes into the initial state, and then you can also stimulate. Yeah? And we observe similar things now looking at Rosenblatt and Pegasus. Yeah, but it's still hard to. So my feeling is still that you have to wait too long for. So if you think about multiple treatments, you stimulate first, and then you have to wait a long time till you really can have a second stimulus that gives you the same high amount. But you can do it the other way, that you pre-stimulate with a low dose and then go for the high dose, and then you get, at least for some time period, a higher response of the system. Yeah, and yeah, but if you ask about interferon specifically, I think the answer is 2 or 3 days, and for Pegasus it's longer, but I cannot say you currently what the value would be. So I have to ask my bachelor student about this. I think he knows, but yes.
Okay. So another audience member asked, a dose response is typically modeled as a Hill function. This response looks different. Is this kind of response observed in any other physiological processes?
Yeah, so in our ODE model context, those responses are not always modeled as Hill kinetics. So let me go to this, whether I have something where you see this. Where is the, sorry, here. Okay. To be honest, okay, this plot that you see here, it's just some sigmoidal regression. So, and you can think of Hill kinetic there. But this was just used for visualization of the data and defining low and high dose for the project. But then later, you really use this this pathway cartoon, and the dose response is shaped by this binding reaction and the degradation of this active receptor complex. And already by this, you get such a typical curve that you can also model with the Hill kinetic. And this sense, and the parameters there are identified. Yeah.
So Western blot data is not that quantitative. Could you perhaps talk a bit more about how this data was used in the model?
Yeah, that's true. So this is always also a little bit the problem. I mean, it's a very cheap technique. Yeah, this is also the thing why it's probably also still used a lot in academia. But it's not very quantitative. Yeah, but so we have the luck that our collaboration partners in Heidelberg, they are really strong in Western plots, so to say. Yeah, and what they really get out, you would call more quantitative than maybe other groups perform. Yeah, so what we, this is maybe also interesting. Let me go to this backup slide there. What we do with it, and this was essentially also published in this paper from 2022, which is called Blotted Optimal Alignment of Western blot data, to get more quantification or to get more data interpretability. We scale this data together. Yeah, so we have different Western blots experiments, and we do lots of repetitions. And then we estimate the scaling factors between these Western blots by means of a scaling model, including also an error model. And then this is what you see on the right. This data has one and the same scale, and the interpretation of the data is much simpler, and you directly see that how big your confidence intervals of the data points are. And yeah, and for at least for lots of our projects, it turns out that it's enough. Yeah, but also with our collaboration, we are more and more also using other measurement techniques, especially mass spec and phosphoproteomics. And with this, we are also hoping that we can identify and model a lot of more stuff. Yeah, so a lot of more reaction networks. Yeah.
So as part of your model development, you looked at parameter identifiability. Can you talk a bit more about how many of the parameters were identifiable or not identifiable in your model?
Yeah. Unfortunately, I don't have this profile likelihood plots at hand, but I can say, so out of these 85 dynamical parameters, it's about 65 that were identifiable, that really had a finite confidence interval. And the others, they were at least not structurally identifiable, so only practically, which means so structurally would also always mean that you can basically fix them somehow or do a parameter transformation to fix it. This we usually avoid, but the practicals remain. And if we can use the model reduction technique that I also showed. Yeah, I can maybe go back to this again. Sorry. Here, we use the model reduction strategy, but if this reduction of the model is in a biological meaning not so meaningful, yeah, or in a biological sense non-meaningful, then we usually don't do it. So then we just live with that and see, okay, we have learned this parameter, we cannot identify, but the essential thing is then, and this is done with the prediction profile likelihood, whether your predictions of the model are still accurate, yeah, and precise.
So in your model, you were using the area under the induction curve as a readout. Do you mean that if we're looking for a patient optimal treatment, we should aim for a higher interferon alpha downstream induction and for a longer period of time?
Yeah, this which measure we use was always also in a discussion point, of course, in our project. We decided for 4 hours in this case, because when you go to this, actually you don't see it that well here because it's only the first stimulation. I don't have plots for the second stimulation, unfortunately, but essentially looking at after the second stimulation, after 4 hours, the main things have happened. Yeah, and this is why we decided to go for this 4 hours. And if you plot it for 6 or 8 hours, the situation doesn't change that much. We also tried this out, but going into the more the clinical setting, it's still a question which readout to take. That is, I wanted also to mention with that the readout matters, but still, let me go one further. Here in this, in this paper, where they linked the RNA to this ISGF3, and you see that if you look one step downstream to what you actually want to have as an effector function readout, it's even different. So it's not only the ISGF3 concentration or the level that you have, but it's there is some still non-linearities at this part, and this you also have to take into account, and this is also what is planned. Yeah.
Great. We do have a bunch more questions, but I don't think we are out of time. So just for our audience, we will be passing these questions back on to Marcus directly. So he can evaluate them and perhaps contact you about the questions.
Yeah.
With that, Marcus, thank you for a very interesting webinar, and I think I've learned an awful lot about how models are handled and some of the output from interferon. So I'd like to say thank you again, and I hope that our audience will come back for another webinar. Thank you, everyone. Have a great day.
Thank you, Rebecca.
Bye, everyone.